Hello, everyone, and welcome to our third and final section of this unit. This section is going to be on meiosis, and meiosis is really the creation of sex cells, sperm and egg cells. So there's a few other things we're going to talk about in this section, dealing with like sexual and asexual reproduction, talking about diploid numbers, talking about fertilization, talking about fraternal versus identical twins. So there's a fair amount of slides here, but we'll try to keep moving through and get you the information we need. Remember, you can always pause as you're filling this out. So when we talk about sexual versus asexual reproduction, when we were talking about mitosis, some organisms can reproduce by themselves. We call that asexual reproduction. So when it's asexual reproduction, there are one parent that is involved, and genetically the DNA is identical to the parent. Okay, so the new offspring have the exact same DNA. They're clones of one another. Some of the advantages, and you should know advantages and disadvantages of each of these types of reproduction. One of the major benefits or pros of asexual reproduction is it's quick, no mates are necessary, and it uses less energy. Okay, so survival of a species, it's quick, you don't need a mate, they're going to live on. One of the cons, or one of the things that aren't good, is there's little genetic variation, which makes them more susceptible to disease. So if there was a particular type of disease that came through and there was, everyone was exactly the same, there are no random mutations that allow for resistance of this disease, the organism is likely to get wiped out. And we see that in society with monocultures of crops, bananas, corn, things like that. Some organisms that go through asexual reproduction is bacteria, some types of plants, and then some like funguses. Okay, asexual mitosis, identical, little genetic variation is what we're talking about. When we talk about sexual reproduction, we are a product of sexual reproduction. Sexual reproduction requires two parents, your mom, your dad. Sexual reproduction is, the process of meiosis was used to create the sperm and eggs that were combined to make us. When we talk about sexual reproduction, we are genetically different from our parents. We have similar traits, but we are different. One of the major benefits of sexual reproduction is we have genetic variation within a population, and we'll talk about why that's important next. When we talk about sexual reproduction, one of the cons of sexual reproduction is you need a mate, and it takes longer. Okay, lots of organisms in the animal kingdom go through sexual reproduction. So I said we talk about genetic variation. What are the benefits of genetic variation? So genetic variation is when organisms are different from one another. You are not exactly like your mom. You are not exactly like your dad. You have similarities, but you're not genetically identical. This increases the likelihood of the survival of a species, not of an individual, but of a species. So the human population, the fish population. Survival of a species. You're going to want to know some of these because you'll be expected to answer some of these talk about some of these on your test. Another benefit is can allow harmful mutations to become less likely. We have mutations in the gene pool, and if it's a bad mutation with genetic variation, it's less likely to become prevalent. Okay, so those harmful mutations kind of get weeded out. And it allows for beneficial mutations to become more common. Those good mutations are going to be more likely to survive and get passed on to the next generation. Genetic variation is through sexual reproduction. Internal fertilization is not required for sexual reproduction. This is saying sexual intercourse is not required for sexual reproduction. For example, fish. They lay their eggs in the bottom of a stream. The male comes and spreads sperm over the top. That is not internal fertilization. That is external fertilization. We often think of sex or sexual intercourse as what is required for sexual reproduction. No, internal fertilization is not required for sexual reproduction. Why does meiosis occur? Because that's what this section is about, is to produce sex cells, sperm and eggs. And why are we making sex cells? So sexual reproduction can occur. So we need to talk about chromosome number. We, as humans, have 46 chromosomes. We've talked about that. But different species have different numbers. For example, fruit flies only have 8, and a fern has 1,260. So it doesn't mean they're more evolved or anything. It's just a number. Sometimes they're bigger chromosomes, sometimes they're smaller. Half of the chromosomes come from the male parent, the dad, and the other half comes from the female, your mom. There are 2 sets of chromosomes, and we call those homologous chromosomes. We talked about that already. Homologous chromosomes, and what that means is that chromosomes, meaning the chromosomes from the males match the chromosomes of the females. We have 2 chromosome number 1s. We have 2 chromosome number 2. One came from your mom, one came from your dad. So when we talk about chromosome numbers, we've already kind of introduced this term, but diploid, a cell that contains both sets of homologous chromosomes, is said to be a diploid cell and is represented by 2N. Body cells are diploid cells. Your skin cells, your hair cells, your muscle cells, your bone cells. Sex cells contain a single set of chromosomes, or we call that a haploid, or half haploid, half haploid. And that's what we talk about with sperm and egg cells. So we said the fruit fly has 8 chromosomes. That's a diploid. So what would their haploid be? They'd only have 4. We as humans have 46 chromosomes, so the haploid number is 23. Chimpanzees, close genetic relative to us, 48 chromosomes. Their haploid number would be 24. Dogs, 78 chromosomes, so their diploid number is 78, where their haploid isn't. This is a picture of a human chromosome, or what they call a karyotype. This chromosome came from your mom, this one came from your dad, this one came from your mom, this one came from your dad. And they can take pictures of these and see what they look like. But notice there's different sizes, and then there's different like banding. So this is a homologous pair. This is a pair of homologous chromosomes. So causes of genetic variation within sex cells, there's 2 kind of different ways that we get genetic variation when we're producing egg and sperm cells. So genetic variation, remember, is important for survival of a species. And this genetic variation is you making the offspring more unique than the parent, so genetic differences. Again, 2 ways. There's something called independent assortment. An independent assortment is when the homologous chromosomes line up in meiosis. They're going to line up randomly in the middle of the cell. So these are homologous chromosomes. In this particular picture, it goes blue-red, and over here it went red-blue. Okay? The fact that this went blue-red and this one went red-blue is random. It's independent. And the fact that this went blue-red and then the next homologous chromosome went blue-red, they are independent of each other. So the fact that this went red-blue is independent that this one is blue-red. This pair is independent of this one. They are randomly assorted in the middle. The other thing we can talk about is for genetic variation is we can get this thing called crossing over, where parts of the sister chromatids or chromatids actually swap during meiosis, and we'll show you that right here. So crossing over is, here's a homologous pair of chromosomes. There is a certain section right here that what happens is, notice right here, blue and red, they are crossed over and then they swap, right? So these swap spots. It's called crossing over. So when this chromatid divides, this chromatid is now different than this chromatid. This one is genetically different from this one. This one is genetically different from this one. This one is genetically different. So we have 4 genetically different chromatids now instead of identical sister chromatids. So phases of meiosis. You should know the phases of meiosis. Meiosis is the process of cell division which we go through, and we are cutting the number of chromosomes in half to produce sex cells. Meiosis, we start with 1 diploid cell and we end up making 4 haploid cells. 1 diploid, 46 chromosomes in humans, will create 4 sperm or egg cells with only 23, half the number of chromosomes. In meiosis, we have 2 separate steps, or 2 separate divisions. We kind of just call them meiosis 1 and meiosis 2. So phases of meiosis. Just like in mitosis where we have PMAT, same idea. We have PMAT, we just have PMAT twice. So we start with meiosis 1, starts with prophase 1. Very similar to mitosis, where the nucleus is going to dissolve. And each chromosome pairs up with corresponding homologous chromosomes, which we refer to as a tetrad. So tetra means 4, 1, 2, 3, 4. There's 4 chromatids. This is when crossing over can occur. Okay? Crossing over was this. Remember when we swap genes, that can occur during prophase 1. Nucleus dissolves, form tetrads, maybe crossing over. Then PMAT, metaphase 1. We have these spindle fibers, once again, that are attaching and pulling on these, and meta-middle. The idea of this is a slightly different now, but instead of all the chromosomes lining up in the middle, now what's happening is the homologous chromosomes line up in the middle. Okay, so the spindle fibers attach to the chromosome and they line up individually and randomly in the middle of the cell. Meta-middle, but homologous chromosomes line up in the middle. See, they're matched up. Those are homologous to each other. Ana apart. Now this time, homologous chromosomes pull apart. Okay, anaphase 1, homologous chromosomes pull apart. We expect you to know these phases. Then telophase cytokinesis, very similar to mitosis, but now what happens is the nucleus starts to reform. So we start to get this nucleus starting to reform, and we get this pinching of the cytoplasm. At the end of meiosis, this is the end of meiosis 1, we have a haploid cell. We have 2 haploid cells. They have 1, 2, 1, 2. There's half the amount. We started with 4. Then we go into meiosis 2. PMAT occurs again, but now we call it prophase 2. Cells immediately go into prophase. They don't replicate DNA. It's already been replicated. It has the exact same steps as mitosis 1. It's just now we have half the number of chromosomes as we originally did. So prophase 1, nucleus dissolves, spindle fibers, all that good stuff. Meta-middle. The chromosomes go to the middle, meta-middle chromosomes, because of spindle fibers. Metaphase 2, meta-middle. Chromosomes go to the middle. Anaphase, ana apart. Sister chromatids pull apart in anaphase 2. The sister chromatids pull apart in anaphase 2. Telophase, cytokinesis. The idea is, once again, they're going to pinch. We're going to get a pinching of the cytoplasm. We're going to start to reform the nucleus. But now when we're done, we have 4 haploid cells that are genetically different. That's important. One diploid cell ends up making 4 genetically different haploid cells. So gamete formation is what we talk about. These haploid cells are gametes. Gametes is another word that we can use for sex cells. So males make sperm haploid cells and females make eggs, which are haploid cells. During the process of sexual reproduction, 2 sex cells come together, and then a haploid plus a haploid creates a diploid. Fertilization, 23 chromosomes combine to make 46 chromosomes, which is 23 pairs. We call that a zygote in humans. We can say the diploid number is restored and will produce a diploid organism. You are a diploid organism because 2 sex cells that were haploids combined to make a diploid, which then developed into you. Now, when we talk about organisms, we sometimes get some unique things that happen, and we get twins. So twins, there's 2 different types of twins. There's identical twins and something called fraternal twins. There's differences. In identical twins, the babies, the twins are identical to one another. They are genetically identical. They're basically clones of one another. Their DNA is genetically identical. And the reason for that is there is 1 fertilized egg combined with 1 sperm and 1 egg combined to fertilize. And 1 egg is going to divide into 2 separate cells that will develop independently. So 1 egg sperm, they got divided and they are genetically identical. Because they're genetically identical, they will be the same gender. They will be the same gender because they are genetically identical to one another. The difference between genetically identical twins and fraternal twins, when we talk about fraternal twins is different. There were two different eggs that were fertilized. There were two different eggs that were fertilized by two different sperm. They are genetically different to one another. They are no different than any other brother and sister are genetically. They just happen to be fertilized at the same time and grown at the same time and developed at the same time. They just happen to have, that has happened to occur, but they are genetically different. It's possible that they could be different genders. It's possible that they could be the same gender. Just as likely that they are the same gender as you are the same gender as your brother or sister. Again, they just happen to occur at the same time. So, big takeaway, mitosis versus meiosis. Mitosis results in the production of two genetically identical diploid cells. Mitosis, two diploid cells. Meiosis, four haploid, genetically different haploid cells. Mitosis, one cell, two cells. 46, diploid. Meiosis, 46 ends up creating four, 23. One diploid cell creates four haploid cells that are genetically different to one another. If you have any questions, please let us know. There's a lot in this section.